Hello, my name is Ron Schwessinger from the University of Oxford, and I'm going to talk to you about dissecting the grammar of chromatin architecture using megabase scale DNA sequence with deep neural networks and transfer learning. We need a very simplified introduction to chromatin architecture to get us going. And when we look at the genome, we usually visualize it in a linear way, in a linear view on a genome browser of some sort, with our genes and enhancers, and maybe we also highlight boundary elements that are frequently occupied by CTCF proteins and are occupied in a convergent orientation, which will become important in a second. Now, this obviously does not quite reflect the structure of the nucleus, in which the chromatin architecture dictates how enhancers and promoters are interacting with each other. To measure these interactions, we need some sort of assays, and proximity ligation assays such as Hi-C are very popular at the moment. And what these proximity ligation assays are doing is basically measuring in a population of cells how frequently are two windows interacting with each other. And they do that, for example, with Hi-C genome-wide. And when you visualize that on top of your linear genome, you can see that triangular structures or domains are emerging that show us which parts of the genome are much more frequently interacting with each other than with the rest of the genome, reflecting what's actually happening in the nucleus. We have enhancers and promoters interacting in insulated domains. If we zoom out to look at a couple of megabases at a time, we still see triangular structures, and we see them at various different sizes. We have bigger and smaller domains, and there's actually a hierarchical pattern to them in that we have smaller domains inside bigger domains, and some domains interacting with each other more frequently than with, again, other domains. Now, we call those domains topologically associated domains, or short TADs, and we call the hierarchical structure meta-TADs, sub-TADs, insulated domains. This really depends on the scale we're looking at and on the author of a particular study. Now, CTCF, this protein, we still find in convergent orientation enriched across the entire genome at these boundary sites. So if you now think about any predictive task, where you just want to predict the 3D genome structure, you could think that you can just join up the convergently oriented CTCF sites and you're done with it. Job done, good job. But unfortunately, there are tens of thousands of CTCF sites in the genome, and their specificity is all but clear. And they're also not the sole driver of chromatin architecture. So how did we approach this problem? We asked how much of the structure is actually predictive from the DNA sequence itself. And why did we think we could do that? Is because we know that we can predict chromatin features very well. Chromatin features meaning things like chromatin accessibility, histone modifications, transcription factor binding sites, and so on. And what we usually do is we take about 1 kb of DNA sequence, and we use, for example, convolutional neural networks with max pooling and ReLU, and a whole compendium of chromatin data, such as from Encoder roadmap, and then predict the probability of 1 kb sequence to be associated with all of those features. Now, what we wanted to do was actually transfer that to the chromatin interaction setting and use high-resolution Hi-C data to train them. Now, the first thing we wanted to do was actually use a large chunk of DNA sequence, at least a megabase, because these domains, these TADs, can be a megabase or even larger. And to fully capture the chromatin interactions, we really wanted to incorporate the entire sequence window, the entire sequence context. Why is that? Because there have been window-to-window-based prediction approaches that obviously neglect the chromatin context. So if you think, for example, that you want to predict the interaction of window A in the genome with window B in the genome, if you would just look at the sequence of A and B and try to predict the interaction frequency, while ignoring that there might be an insulating element in between them, you probably not get amazing predictions. So we really wanted to take the full context as much as we could into account and look at the bigger picture. Now, the first trick that we employed was transfer learning, because we hypothesized that actually the sequence patterns that we can learn and that are predictive of chromatin features, such as CTCF binding, will be useful for predicting chromatin interactions. So in the first training phase, or first training step, we train a convolutional neural network to predict chromatin features, such as CTCF binding and transcription factor binding, as we've been doing for 5 years. And then use the convolutional weights of this network to transfer them and seed the convolutional filters in our chromatin interaction network. What we then do is we follow these convolutional layers with max pooling up by dilated modules, so several dilated convolutional layers, which are just really good at aggregating a large spatial context, a large sequence context, without you needing to do max pooling and lose the resolution. Follow that up by a fully connected layer, and you can predict chromatin interaction frequencies. And we train these networks then using cell type specific Hi-C data. And at the moment, we train one model per cell type using tissue specific Hi-C data. Now, how do we encode the Hi-C data? Well, we take about a megabase of DNA sequence. We chunk it up into genomic bins, for example, 5 KB bins, if we want to have a 5 KB resolution model in the end. And then we encode the interaction frequencies as sort of a vertical pole in the middle of that DNA window, where every number in our target vector would then be the interaction of bins with other bins. For example, the first number would be interaction of bin 1 with itself, then we have bin 1 with one bin upstream, and walking our way up like this, so that the last number in our target vector is actually the interacting of 2 genomic bins that are 1 megabase apart. And we're ignoring anything longer than 1 megabase in our models currently. Now, the second trick that we're doing is we're normalizing the Hi-C data to get rid of the distance dependent signal. So if you look at raw Hi-C data, you can see that the interactions that are closer to the bottom, that means that they're closer in linear space in the genome, have a much higher interaction frequency than anything that's further apart in linear space. And this is simply a function of the DNA as a polymer. So if you think of a rope or a string, any 2 points in the DNA that are very close in linear space will be interacting much more frequently, no matter how you fold this rope or the string up in 3D space. Now, in a way, this is not really what we're interested in. We want to know what other signal is there on top of that proximity signal. So we do a distance stratified normalization and do actually a percentile-based normalization, where we put all those interaction frequencies into separate bins and use a little trick in that we do put lower interaction frequency counts into smaller bins and to bigger bins, and put high interaction frequency counts into finer grained smaller bins. So what we get out of this is that we basically get rid of the proximity ligation signal of this gradient in the Hi-C map, and we also highlight domains and the domain structure a bit better. This is now how Hi-C data actually looks like. So we've got B-cell Hi-C data, and you actually at 5 KB resolution need 3.6 billion reads to get to Hi-C data as highly resolved as this one. But you can see there's this gradient signal, so you have much more interaction frequency at the bottom, closer in genomic linear distance. And then you see triangular structures at different sizes and in various hierarchical forms with smaller and bigger domains interspersed. This is how the same data looks like when we applied our skeleton transformation. So you see that the gradient signal is gone, and we also highlight these domains better than we do in the normal Hi-C data. And this is finally how our model, after being trained on several chromosomes, predict on a holdout chromosome. So only taking a megabase of DNA sequence and moving from upstream to downstream along the genome, basically. So you see that we get, first of all, much smoother predictions than the skeleton or the Hi-C data look like. And the model actually captures larger domains, captures smaller domains, and captures hierarchical ordering inside these domains as well. And we can get that to work on different resolutions and across different cell types and Hi-C data depths, so you don't need a crazy amount of Hi-C sequencing to do that. In a cross-validation scheme across chromosomes, we get a correlation between a skeleton and a prediction of about 0.45 across the different chromosomes. Now, this doesn't sound amazing to begin with, but when you compare it to other methods that actually use CTCF binding from ChIP-seq or other histone modifications directly, they get about the same correlation values. Well, we have now a sequence-based model to work with. What can we do with that model? Well, first of all, we can start to fine map Hi-C data, which is really interesting. So 3.6 billion reads is a lot of sequencing to go into. From the same study, there's also K562 data, which only has 1.3 billion reads. And when you try to visualize that at 5 KB resolution, you see that the domain structures become suddenly a lot less clear and more faint and sparse. But you can still train a deep neural network to predict the chromatin interactions based on those sparser data set, and you get really nice, smooth chromatin predictions. Now, what you can do then is actually use algorithms to call boundary elements, for example, or call domains and call these boundary elements. When you employ that on the high C data itself, which is much sparser, you get sort of an overcalled domains with potential boundaries being placed all over the place. Whereas if you use the predictions, you get a much better, finely resolved domain call. And if we then use another proximity ligation assay called TileC, which is doing the same thing as high C, but just on a focused window so that you can focus all your sequencing efforts on a couple of megabases, getting a much higher sensitive view of your locus, we can see that these predictive domains much better line up with actual domains in the structure and in the chromatin architecture. The other thing that we can do is we can start to map the sequence determinants at base pair resolution. So you're probably all familiar with saliency scores in various shapes and forms. And when we compute the saliency, this important score of the linear sequence for the interaction predictions, for example, in this Icarus 2 locus over here, and overlay that with CTCF, ChIP-seq, open chromatin, as well as some histone modifications, we can see that we get sharp peaks of this saliency score around CTCF sites, which is really reassuring because we know that CTCF plays a major role in chromatin architecture. When we zoom in, we can also see more broad patterns, more broad peaks over promoters and of the saliency score. And we zoom into the base pair level, we can see that we can recover or rediscover the CTCF motif as well as transcription factor binding motifs and promoters. This is actually a genome-wide pattern that we see. We see really sharp peaks at CTCF sites and more broad peaks underneath promoters, especially active promoters. Now, why would we care about a model like this? Because we can do predictions of the impact of variance on genome architecture with a model like this. For example, structural variance. Using an example from Hinz and colleagues from 2016, what they did is using CRISPR-Cas9, delete a 30 kb fragment that contains 4 CTCF sites, and then using 5C, which is just another proximity ligation assay as far as we're concerned now, showed that when you delete those 4 CTCF sites, this chunk, you can observe that you have 2 domains in the wild type, and then upon deletion, you get a merging of those domains into a broader, bigger domain. Now we can recapitulate that effect when using our sequence-based model, and that we see that the model in wild type predicts 2 domains and a merged domain upon deletion of that particular sequence. Now, structural variations you could do with polymer physics models and stuff like that earlier, but what's really exciting about the sequence-based model is that you can actually now use it to predict the impact of single nucleotide variations. For that, to demonstrate that, here's another example from Schmiedel and colleagues, 2016. They have identified 2 SNPs that are associated with asthma risk and forming a risk allele and a non-risk allele. Basically, using CTCF binding as well as motifs, they hypothesized or demonstrated that these SNPs are likely to disrupt a CTCF binding in the risk allele and create a CTCF binding in the risk allele relative to the non-risk allele. Now, if you put those SNPs into the reference genome and only those 2 SNPs, and then predict the chromatin interactions with DeepC, we can see that on a different scale with smaller fine-scaled color spectrum now to get to the minute differences. We see that actually the leftmost SNP here, or underneath those leftmost SNP, you get an insulation effect appearing in a non-risk allele, whereas in the risk allele you have much more interactions, a much more homogeneous domain. It actually fits really well with what the authors hypothesizes that the OMDL3 promoters interacting with intronic enhancers from Icarus 3 in the risk allele, matching to this more interaction present domain. The other thing we can do is we can do genome-wide deletion screens, for example, where we actually just take every single enhancer, promoter, and CTCF site across the genome and delete them one by one, predicting their effect. And when we do that and plot the average effects of those sites, we see reassuringly again that CTCF sites, especially the ones strongly bound by CTCF, have on average a much bigger effect on chromatin architecture. But what's also interesting is if you compare promoters to enhancers, it appears that promoters, especially the ones strongly active, marked by active histone marks, that have on average a bigger effect than do enhancers. So this brings me to the summary. So I hopefully convinced you that the 3D chromatin architecture is somewhat predictable from DNA sequence. that a sequence-based model that we have allows us to map the determinants of chromatin folding at base pair resolution, and that we can use this model to predict the impact on chromatin folding from single base pair variants and structural variations. This just leaves me to thank my labs and everybody who was involved and helped in that project, and point you to our preprint. And the manuscript has been in principle accepted by NHMF as we speak, so this should be in print when you see this presentation. And also I should have a link over here. So thank you very much and looking forward to your questions.